Well, good afternoon. And it's really my privilege to introduce to you today's speaker, Dr. Effie Petersdorf. Effie is a professor in the Division of Translational Science and Therapeutics in the Department of Medicine at the University of Washington School of Medicine. She's also attending physician in clinical bone marrow transplant services at the Fred Hutch Cancer Center. She holds the Madeline, Dabney, Adams Endowed Chair in AML Research at the Fred Hutchinson Cancer Center. Dr. Petersdorf earned her bachelor's from Harvard and her MD from McGill University. She completed her residency in internal medicine and fellowship in oncology at the University of Washington before joining the Fred Hutch Cancer Center. Dr. Petersdorf is an accomplished physician, scientist, and educator. She's received numerous honors and awards, and just to name a few, President Bill Clinton awarded Dr. Petersdorf the Presidential Early Career Award for Scientists in Engineering in 1999. She then went on to be the recipient of the Cipollini Award, a very prestigious award from the European Federation for Immunogenetics, the 2018 Hillier Festenstein Lecture of the British Society of Histocompatibility Immunogenetics, and the 2019 recipient of the Rose Payne Award from the American Society for Histocompatibility Immunogenetics. And then finally, just recently, the Lifetime Achievement Award from the American Society of Transplantation and Cellular Therapy. She's past president of the World Marrow Donor Association and the American Society for Blood and Marrow Transplantation. Effie has led training in immunogenetics and stem cell transplantation for countless trainees at UW. And she's published extensively in high-profile journals and has had numerous grants supporting all of her research. Dr. Petersdorf has devoted her career to determining how genetic factors influence the success of stem cell transplants. She's pioneered molecular methods to understand the impact of HLA matching on stem cell transplant outcomes, including risk of graft versus host disease. More recently, Dr. Petersdorf identified ligands regulating T cell and NK cell alloresponses, including polymorphisms residing in non-coding regions of HLA-B, DPB1, and HLA-DQ alpha beta heterodimer structures, as well as MIC-A, MIC-B ligands with NKG2D. This exciting output, this phenomenal output of work by Dr. Petersdorf's research has directly changed clinical care and the practice of immunogenetics in the field, especially in optimizing selection of allogeneic donors and improving transplant outcomes. So I just want to thank Effie so much for visiting with us today, and we can't wait to hear about your exciting work and look forward, of course, to your presentation. Thank you so much.
Thank you very much, Dr. Reed, and to all of you. Thank you for the invitation to join you, UCLA, the CTSI seminar series. Something probably needs to be down there. Yep. That sounds better. For me to come to UCLA is an absolute special treat because, of course, HLA and solid organ transplantation started right here at UCLA. Pioneering work of Dr. Paul Terasaki, who, when he chaired the very first workshop at UCLA, he was really already inspiring all of us in the community with collaborative research. So he was the one who really got out serological kits to participating laboratories so that everybody could be involved. And he was such a master, a scientist, a physician, clinician, that he not only chaired one HLA workshop, but he went on to chair a second workshop in LA, 1980. And this was a very special workshop because the 1980 workshop showcased the outstanding outcomes of the UCLA kidney transplant registry data and showed for the first time very pronounced effects of HLA matching and mismatching on kidney allograft survival. And so to me, this really sets the stage. When I think about HLA and transplantation, it is most definitively HLA and solid organ transplantation that started right here at UCLA. And in fact, this 1980 workshop was the first workshop where laboratories were struggling with being able to identify what these locally named specificities that they coined MT and MB. And it was after this Los Angeles workshop that the nomenclature committee got together and formally officially named MT and MB HLA-DQ. I wish I had been here in LA in 1980 because I'm going to be showing you some new work that we have done in HLA-DQ. But really the template that Dr. Terasaki established really provided so much enthusiasm within the community that when the late Dr. John Hansen was awarded chairmanship for the 13th International HLA Workshop held in British Columbia and then the Congress in Seattle, we said, well, we must have a component or a working group, HLA in hematopoietic stem cell transplantation. You are already very much ahead of the game for HLA and renal, but not so much in blood and marrow, where we were still in a way in its infancy. So we were very proud in the 13th International Workshop, we had managed to pull together a collaboration that resulted in about 900 patients who had undergone an unrelated donor transplant. But this really created so much enthusiasm. We now have a wonderful international collaborative effort, spans transplant centers, transplant registries, donor registries, immunogenetics laboratories, and data scientists. And now for the upcoming workshop in Japan in 2026, we have a data file for over 70,000 patients who've received a transplant either from a volunteer, unrelated, a haploidentical, or a cord blood unit. And this is probably one of the most important slides, is to acknowledge the contributions of the international community, the registries all around the world. So we have Australia, the UK, the UK, throughout the United States, Europe, Japan, and the regional registries within Europe. Well, the data that I'd like to share with you today, much of it stems from this working group, the International Hispanic Working Group. And we began with asking fundamental questions and hypotheses in the volunteer unrelated donor scenario. As you all know, because of the growth of international donor registries as shown here from the World Marrow Donor Association, WMDA, we've really enjoyed a tremendous response, a humanitarian response. We have 42,470,000 volunteer donors and cord blood units worldwide that are there ready to serve for blood and marrow transplantation. It's such an unbelievable worldwide response. And this has really catapulted the use of volunteer donors when patients did not have suitable options within their family. But as all of you in HLA know, something else was also increasing, and that is the number of unique HLA variants, such that before, when the WHO, the World Health Organization, began to catalog these variants, we were really sitting at 900 some unique allelic variants. But as of August of this year, we have over 40,000 unique variants. That is a mind-boggling degree of human genetic diversity. And if you think for a minute, how can we encompass that information in our day-to-day clinical practice when we are really searching unrelated donor registries, cord blood banks for that perfect match for patients who need a stem cell transplant? If in fact the worldwide data suggests to us it's important to completely, precisely match the transplant donor, then the IHWG data from the Asilomar 2017 workshop taught us that every additional mismatch, there is about a 7% decrease in overall survival. So we all want our patients to be here, right at the top here of this survival, Kaplan-Meier survival probability, and these reflect 6 locus, ABC, DR, DQ, DP, 12 of 12, or a single DP, 11 of 12. But you can see the deleterious effect of additional HLA mismatching to overall survival. We also know that the probability of finding a well-matched donor depends on the size of the registry, the composition of the registry, the specific alleles, and the haplotypes of the patient's alleles. And we know that for many of our patients of non-Caucasian background, they have a much lower probability of identifying a suitable match for their transplant. Here, this was work by the National Merit Donor Program, CIBMTR, by Lauren Greger and colleagues, that shows really the profound differences in how our patients can find donors. So there's been a worldwide effort, of course, to identify alternative donor sources for patients who don't have that match. And as all of you are very well aware, here is where haploidentical transplantation, the growth here, this green curve of haploidentical transplantation is paralleling the growth that we have seen in the past for unrelated donor transplantation. And what I like to do today is focus on new lessons that we are beginning to learn in the haplotransplant situation with post-transplant cyclophosphamide or PTSI. Now, here are CIBMTR data, and they tell us that yes, cyclophosphamide is relatively easy for physicians to administer. Yes, most of our patients do tolerate this drug, but we are still gaining information and clinical experience in long-term effects. And we already know causes of death after haploidentical donor transplantation in the US, patients transplanted between 2018 and 2020. Primary reason for transplant failure is recurrence of the underlying malignancy, whether or not the patient was younger than 18 years or 18 years or older, 42%, 50%. The two other major causes for failure are organ failure here in this light green, and then infectious complications, 18% and 17%. So we still have much to learn about the HLA barrier and what potential role HLA may have in modulating these effects. So I'm going to present today 3 different models, and I decided that I would take more of a 37,000-foot level view of the MHC, and I'm going to share with you information on non-classical class 1 and the importance of the NKG2 axis. I'm going to delve a little bit into the HLA-B locus and then spend some time on the class 2 region. And the most important lesson that I'm really beginning to appreciate is it is not completely about precisely matching every donor to every patient. We need to really think out of the box and go beyond the match-mismatch model. And what's really fascinating about the new data is that they are telling us that germline effects from the patient and the donor are just as important, and particularly for haploidentical transplants, where you only share one haplotype and maybe variably match or mismatch for the non-shared haplotype. So each of these models will show us that germline is so important in defining risks of relapse and mortality. I'm going to start with the NKG2 axis. So what does this mean? Right in the middle of HLA, amongst your classical class 1 and class 2 loci, you have 2 what we call non-classical genes, MIC-A and MIC-B, and they are 2 of a series of ligands for NKG2 receptors, and their particular receptor is the NKG2D. It's an activating receptor. And we've been really interested in the non-classical class 1 complex because it is inherited side by side HLA. It's in fact very strongly linked to HLA-B, but it's been a very difficult group of loci to analyze because of that strong LD, linkage disequilibrium. So that if you were to take a very well-matched transplant population and try to look at even patient-donor mismatching, you will find that the vast majority of HLA-matched transplants are also MIC-A, MIC-B matched. NKG2D is expressed by NK cells, cytotoxic alpha beta CD8, and gamma delta T cells. So it sits at the perfect intersection of the NK and T cell pathway. And for this reason, we were very interested in really tackling the question of MIC-A, MIC-B, and NKG2D in the haploidentical scenario. Its major function is to survey the landscape for stress cells, which are either infected or have been transformed or cancerous. And so this served as a very nice initial model for understanding relapse and transplantation. Now, MICA was a locus that has received a lot of attention by laboratories in the clinic. And for good reason. There was a lot of basic research in the 1980s that really shed light on MICA. And a lot of the literature that you'll find in stem cell transplantation has focused on a very curious dimorphism at residue 129, where either a methionine or a valine can be found. This sits right here in the alpha-1 domain, so you can see the extracellular portion of MICA that is really within direct contact with your NKG2D receptor. The MET-VAL dimorphism is right here in the alpha-1 domain. And the reason why a lot of laboratories really focused on this residue 129 is because Alexander Steinle and colleagues over 20 years ago made a really important discovery. They taught us that this methionine valine switch in the alpha-1 actually changes the affinity of MICA to its NKG2D activating receptor. So of course, this spurred a lot of interest to say, if you have a differential affinity of the ligand to the receptor, potentially that could be meaningful functionally in transplantation. Well, there were a lot of papers that ensued looking at MICA 129 methionine valine. Here's a really nice paper from the Carapito team in France. Although they showed some sort of a correlation with acute GVHD, this did not translate to either higher or lower probability of survival. A second study from Europe by First and colleagues took a slightly different approach. These are all unrelated donor transplants, principally cyclosporin, TAC, calcineurin inhibitors for GVHD prophylaxis. And what they did was they asked the question, if you integrated MICA into the schema of donor matching with other classical, what do you actually find? And here they were able to show beautifully that MICA seemed to function as a classical transplantation determinant. Something very different, however, in the United States, Medhar Asler performed a study on behalf of the CIBMTR in US transplant centers, and we did not see any notable correlation of MICA with clinical outcome. And then the last study was a SNP discovery study by Paul Martin and John Hansen at Fred Hutch. And they found something even more different, that it was really the donors MICA 129 dimorphism that truly mattered for non-relapse mortality. So this is the setting in which we approached our study. And we were very curious because all the studies seem to focus on the alpha-1 domains residue 129. In fact, MICA is a very interesting molecule. It's right here, exon 5. You have a very curious phenomenon. There is a short tandem repeat, or STR. And this short tandem repeat changes the stop, this transmembrane part of MICA. And this, in fact, is so critical in the amount of MICA that can be expressed. And that was shown by Mizuki and colleagues from Japan. This STR can insert anywhere from 4 to 10 different alanines all in a row. And this happens right in that transmembrane stop. But then on top of that, there is a SNP that occurs only on certain haplotypes. And there is a single base insertion that when it's present, it's a premature stop codon. And what Mizuki and colleagues showed was that, in fact, between the STR and the SNP, you could really alter the ratio of how much MICA was expressed on the cell surface and how much was free as a soluble molecule. So we decided perhaps the answer to MICA was really in understanding the entire molecule, not just one part of it, either the alpha-1 domain or the transmembrane portion. And we set up a retrospective study, 1629 patients who had received a haploidentical related transplant in a US center. Most of the patients, the vast majority, did receive post-transplant cyclophosphamide to prevent GVHD, and most patients had a malignancy. They were predominantly AML, ALL, MDS, but we did have a few patients who received transplantation for a life-threatening non-malignant disorder. And what we did was we just methodically said, let's just really understand MICA, not just one particular residue, but the entire protein, the entire molecule. And so we sequenced through MICA and characterized the methionine 129 the exon 5 repeat, and that all-important stop insertion. And then for McB, there is almost nothing in the literature on this molecule. And so we decided to take a very focused approach and tackle the 4 missense residues. For donors, we looked at the NKG2D receptor, and I'll get a little bit into this, a very interesting residue 72. So for the sake of time, I'm not going to show you the negative data for residue 129. We didn't see any pronounced effect of this methionine valine 129 on any clinical endpoint per se. However, there was a really striking correlation of that transmembrane portion, the stalk of the molecule. And this time, if the donor was mismatched with the patient, that was actually correlated with superior survival. So not really aligning with our classical teaching that matching is always good, mismatching is risky. In this case, it was flipped. Donor mismatching for the transmembrane was protective. But there was a twist to this. That effect, this protective effect, was made even stronger if you knew what was happening in the alpha-1 domain for the residue 129. And what we found is amongst the patients who received a transplant from a haplodonor, mismatched at the STR for the transmembrane portion, but they were matched at residue 29, then this even further improved overall survival. So here you have 2 seemingly independent features of the same molecule, but each has a very important function. On the one hand, your alpha-1 domain is coming into direct contact with your donor's NKG2D receptor. On the other hand, the amount of MICA presented on the cell surface to that receptor and the amount that becomes solubilized is governed by a totally different part. And so clearly you need information across the length of the gene. We were very excited to tackle McB. As I said, almost no information. These are the residues we were interested in, and McB, contrary to opinion, it is polymorphic, and there are more and more alleles being discovered every week. We did not see any influence of donor McB on any transplant clinical endpoint, but we saw a pronounced effect for the patient's germline McB. Aspartic acid, whether it was homozygous or heterozygous, was associated with superior survival compared to the lack of a McB molecule with aspartic acid. And this was the first indication that, in fact, this missense position may, in fact, give us a clue in terms of functionality. Now, the donor NKG2D receptor is fairly well conserved from a sequence standpoint. There is a very curious polymorphism at residue 72. It has no bearing whatsoever. It is nowhere near where this receptor would make contact with either McA or McB. However, this missense residue has a very important function. It is also an expression quantitative trait locus, or EQTL, which means it has the potential to influence higher or lower expression of that receptor on NK and T cells. And in fact, if you look at 1,000 genomes data, you will see that the distribution of this polymorphism really varies quite strikingly between the African, Hispanic, Asian, and North European populations in 1,000 genomes. So we were very eager to tackle this. And we're very surprised to find that transplantation from donors who were threonine homozygous was associated with superior survival. And this is not accounting for patient McA or McB at this point. But this was the first indication that potentially it really isn't the necessarily protein sequence that's making a difference. It's how much receptor is actually present to be able to engage with its ligands. So now we have 3 findings. We have patient donor match mismatch, exon 5 for the short chain repeat. We've got the patient McB residue 52, and we've got the donor NKG2D receptor residue 72. One of these is driven and specified by the patient. That's the patient's McB 52, which we cannot modify. But the remaining 2 are driven by the donor. So if we were to have a patient who had 2 or more potential family members, and they're all haplo, how would we use this information? Can we be smarter and basically pick a donor that would even correlate with higher survival? So that's exactly what we tested. When patients encode the favorable MICB residue 52 aspartic acid, here you can see that if you had the opportunity to choose a family member who was both mismatched for the exon 5 MICA and had an NKG2D receptor that was threonine at residue 72, you can actually improve relapse by 60%. And that is a huge step in the right direction. So I love this model because it tells us that we've got to think beyond just single polymorphisms and SNPs. We've got to take the entire protein, the entire molecule, and importantly, what is the patient presenting and what is the donor offering and how do these come together? Certainly, we learn much more when we combine the donor's receptor with the patient's receptor, much more than either one on its own. And so that's a new lesson for us. I'm going to quickly go to the HLA-B leader peptide because that's another very fascinating story that we stumbled upon because we were searching for how do we define permissive B mismatches for our patients. The B leader was actually inspired by IHWG data. This came out of the Asilomar workshop, 33,982 international transplants, unrelated donors, all of them received some sort of a calcineurin-based GVHD prophylaxis. And here you have the data of 10,415 of those transplants who received an unrelated transplant with a single mismatch at either A, B, C, DRB1, DQB1. And so this model only asks the question, if you didn't have a match, are there certain mismatches that may be more preferable than others? And so we learned that for whatever reason, DQ and DR were associated with far less detrimental impact on survival. But look what happens, particularly for HLA-A and B mismatches. And I put B in red because, as many of you know, in HLA, B is the most polymorphic gene in the entire genome. And as of August, the August IMGT WHO committee report, over 10,000 unique B alleles. How, as transplanters, are we able to accommodate that? We really do need to match for B, how do we do that? If we can't match for B, what do we do? How do we choose amongst B mismatched donors? And that is why we tackled the B leader. So we're all familiar with class 1, 7 exons, exons 2, 3, 4, define alpha 1, alpha 2, alpha 3 domains. And most of the substitutions really are focused in the alpha 1, alpha 2 regions, which define the all-critical peptide binding region. But the peptide binding region and the groove is physically connected on the DNA to exon 1. And exon 1 produces a very tiny 8-9 amino acid long baby peptide called the leader. And sometimes you'll see in the literature, it's also called the signal peptide. And B is very unusual because unlike A and C that are invariant, at the -21 position of exon 1, HLA-B, there is a curious SNP. It's a dimorphism. And so the peptides that are contributed by the HLA-B locus encode either a methionine or a threonine at the second position of this leader or signal peptide. Now, this isn't just for academic reason, out of academic interest. This dimorphism actually plays a very important role in the expression of HLA-E, another non-classical class 1 encoded within the region. And here you have beautiful data. This is from Amir Horowitz when he was in Peter Parham's lab at Stanford. We've also replicated these data. Possession of leader peptides with methionine at the P2 strengthen the cell surface expression of HLA-E, both E0101 and E0103, whereas threonine contributed by B, A, and C are really associated with much lower the expression. So here is a potential that you can take polymorphism that is physically situated in the B locus, but it may have very far-reaching consequences elsewhere for other molecules that participate in both T cell and NK responses. So we decided, okay, you have exon 1, which defines this very important leader peptide, and that is physically linked on the chromosome to exons 2, 3, 4, which define your peptide binding repertoire. So we collaborated with Dr. Colin O'Higgin at the NCI. He is just a world's expert in evolutionary biology. And we asked the question, if you just align the sequences, take 1,000 base pairs of contiguous class 1 sequence, what do the clades, what do the families look like? The clade is basically a genetic term for groups of alleles that are evolutionarily conserved and have a common ancestor. And what's really fascinating is, I hope you can appreciate, you have a very large clade here comprised of many alleles, a smaller clade here. Column anticipates these are at least 8 million years of age. That's when at least the bifurcation came. So you have two different families. But what's really fascinating is that they have preserved their linkage to their exon 1, so that this top clade, every allele has a threonine at the P2 position. And every one, but except with one exception, there's a recombinant allele down here, is connected to an M. So we said, okay, even though they have a connection by LD with exon 1, they clearly represent a clade of alleles that function from the antigen presentation standpoint. These alleles in the T clade may be more similar than these alleles in the M clade. So what happens if you do an HLA-B transplant? If the patients and the donor's mismatched B alleles are both from the T clade, maybe their outcome is better than if the patient came from the T and the donor came from the M, or vice versa. So we decided to put that to the test. And we just used T and M as a surrogate name, leader matched or leader mismatched. And we hypothesized that if the B mismatch was between two alleles in T or between two alleles in M, we called them leader match. And we hypothesized that the risk would be lower compared to if the mismatch occurred between clades. And in fact, this is what happened. Step up of grades 2 to 4 GVHD, at least. We didn't quite meet statistical significance for grades 3 to 4, but I think the trajectory was already there. So clearly we can use the evolutionary, the phylogeny of HLA-B to group B mismatches into those that are more favorable or permissible, meaning leader match, versus those that are less so, leader mismatch. We were able to confirm these findings in a totally independent transplant group. This is a collaborative study from the CIBMTR. It was spearheaded by Ephraim Fuchs and Shannon McCurdy. There was a retrospective cohort analysis of over 1,400 US transplants, haplodonors. All patients received PTSI, and patients either carried the diagnosis of AML, ALL, or MDS. And here we actually saw the same effect in haplo, PTSI, step up of mortality risk, and TRM with B leader mismatching. So these are all haplo transplants. They are all B mismatch, but those that are leader match have better overall outcomes than those that are leader mismatch. So that was a really reproducible validation. Now, this is where we've learned a lot from Dr. Terasaki and the UCLA workshops, because Dr. Terasaki set the example. Collaboration is essential because not any one of us will have the data to really connect the dots for the whole picture. So we reached out to the Be the Match bioinformatics team, and we said, okay, we've got this fantastic correlation. What's emerging is this leader is really looking like it is telling us something clinically. And the question is, well, if you surveyed the entire US population, how polymorphic is the B leader? And so we were very lucky, a little shy of 1.1 million US donors. And they all had next generation sequencing that allowed us to phase exons 1, 2, 3, and 4, leader with the peptide binding group. And what we can say is 99.99% of all of the B sequences either had a methionine or threonine at P2. So we knew we were good, that we weren't having to worry about other polymorphisms that we had to consider. And yes, there are substitutions elsewhere, but they are occurring at a vanishingly low frequency. So 6 examples out of 2.2 million haplotypes. That's very low. So it's not that these are not functional, yet to be determined, but they're occurring at a very, very low rate. The next question we had is, if we're really going to help transplant clinicians and search specialists translate the B leader, we have got to be able to help them accommodate all different types of HLA typing, from the lowest of the low resolution to the ultra high of next-gen sequencing. And so we asked the question, can you rely upon the serological group to define that leader? So this is where I think I would love to have presented this to Dr. Terasaki to see what his reaction would have been. The answer is yes, you can rely on what we would call the first field low resolution specificity. So B7 is part of this red clade M, and almost all, with only 1 or 2 exceptions, will it not be an M, and so on and so forth. This is really going to help, because it means that you don't have to require next generation ultra high sequencing, and you certainly don't even have to sequence exon 1. You can by LD assign the leader based on serology. And so now this tool has been put together to help HLA labs, transplant centers, search specialists. And what you can do is input any level of typing for B of your patient, any one of donors. It could be related donors, unrelated donors, cord blood units. And what the program will do is will give you the leader genotype of the patient, all the donors. It will then do every pairwise comparison of patient with donor 1, patient with donor 2, and it will rank order and offer you suggested rank order of a preferable donor. And so maybe some of you have used this tool. It launched 2 years ago. Really delighted. There's been a tremendous use of this tool. And when I talk to people, they are really integrating it into clinical practice. So if you haven't had a chance, I would love you to take a look at it. And I always love getting feedback, and because we're always trying to improve things. I'm going to go to the last topic in the next 10 minutes. The class 2 region to me is just an enigma, because here you have this huge segment of the MHC, and these loci are basically duplicated evolutionarily. And you have this very curious phenomenon where you've got loci that encode antigen presenting proteins for antigen presentation, but then you have side-by-side molecules that actually facilitate the loading of the peptides into those molecules to present the peptide to the T cell receptor. So DR, DQ, DMDO, and the only locus I didn't put on the cartoon is DP, but they're all what we term a classic alpha beta heterodimer, where the alpha chain is prescribed by A genes and the beta chain by B genes. And so the one next question we had is, we have individual potential locus effects, but now you have all the machinery you need for antigen presentation and how to get the peptide in the groove. What do they all have to do with each other? And is it important that if you know DR, do you know the rest? Or if you know DQ, you can predict the rest? And how does that impact patient outcomes? So we did this retrospective analysis, and we tackled each gene individually. So we took, for example, DRA by itself, DRB by itself, etc. And then we looked at it in patients separately from donors. We then put together the alpha beta heterodimers, look at alpha beta in patients, alpha beta in donors, and then the all critical patient donor match mismatch. I'm not going to show you the negative data for DR alpha. Yes, I was a bit disappointed, but no, it means that I wasn't smart enough to figure out which one to go after. So there's still work to be done with DR alpha. And just because this study did not show correlation, it does not mean that the DR alpha polymorphisms are not functional. We found a really curious association with DR beta, 4,700 unique DR alleles. So the haploidentical PTSI literature has shown for many, many years now, first data from the Hopkins group here on the left, Casamon and colleagues, that haplodonors that are DRB1 mismatched are associated with lower post-transplant leukemic relapse. And this was a really new finding that people said, well, how do you explain that? What is the mechanism? We still to this day are trying to understand how a mismatch can be favorable in this particular clinical scenario. 10 years later, we have an independent validation, DRB1 mismatching associated with lower relapse, PTSI haplotransplants. So when we did the CIBMTR analysis, that's one of the first things we wanted to do. And yes, we were able to validate it. Here you can see 35% lowering of relapse associated with haplodonors who are DRB1 mismatch. So now everybody is very excited because when we choose our haplodonors, we're actually deliberately looking at those who are DRB1 mismatch. And as you all know, because they are haplo, the vast majority of your haplodonors will be DRB1 mismatched. And we ask the question, are all DRB1 mismatches equally favorable? What if you have a choice of a large family where you have different combinations of DR mismatches? Is there some advantage to choosing one DR mismatch over another? And the reciprocal question is, are all DR matches to be avoided? Are there certain DR matches that actually are better? So we pose this question, and the first thing that I did was go back to structural biology. So the brown structure of DR, this is, pretend you're a T cell and you're a T cell receptor, and you're going to land on your DR molecule. So this is what the T cell receptor sees. You see the groove here with the peptide in pink. And here you've got a residue 86. And very interestingly, most HLA DRB1 sequences either have a glycine or a valine at 86. And this dimorphism, even though glycine and valine is not a very drastic change, it has a very profound impact on the pocket, which does accommodate the side chains for the peptide anchor. And so glycine and valine will basically open or narrow this end of the DR molecule. And so is a very important regulator of the peptide repertoire that is selected. So we decided to put this to the test. And the first question is, are all DRB1 matched haplodonors equally favorable? And the answer is no. Here you can see disease-free survival, superior survival in this group of patients who are heterozygous glycine valine, less so for patients who are homozygous glycine or valine. Remember, patient donor are DR matched, so patients 86 equals donors 86. So right off the bat, you know that in fact, there is something going on with the DR, potentially DR peptide, in that there are certain scenarios where a patient who is fortunate, whose germline is a heterozygous glyvale, does enjoy superior disease-free survival. Now we had a much larger group of patients who were DRB1 mismatched. And the question was, are all DRB1 mismatch haplodonors favorable? And the answer is maybe not. Who benefits the most from a DRB1 mismatch haplo? Patients who are glycine, who receive a transplant from a donor who is glycine valine. So this haplodonor glyvale into a glyli patient creates a mismatch valine donor versus glycine patient. And here you can see the protective effect of that mismatch, but we did not reach statistical significance for glyvale or vale. So lessons learned, we now have a really novel, very interesting, simple dimorphism for DR. Now we're going to go to DQ. And DQ, we learned through the unrelated donor experience that the alpha and the beta are critical to understanding function. So here, for those of you who are not as familiar with the structure of DQA1, DQB1, you have two parental haplotypes. Here, one haplotype I've just illustrated in this cartoon. You have a yellow DQA gene with a blue DQB gene. Second parent, you've inherited a green DQA with a purple DQB. And so to construct your alpha beta heterodimers, you have one molecule which is the product of the yellow and blue. That's this molecule. We call that cis because it's on the same chromosome. The other parent is contributing the green and purple. That's this cis. But there are certain haplotypes that the alpha from one parent can connect with the beta from the other and produce, in this case, the yellow, purple, or the green, blue molecule. And that is called a trans heterodimer because you're going between the parental. So now when you think about it, the question is do all haplotypes, can you do the Cartesian product? And the answer is no. And the beautiful research actually came out of the type 1 diabetes work, Jerry Nepom, Bill Clark from Virginia Mason in Seattle. They actually show that only certain DQ alphas will form a stable heterodimer with certain DQ betas. And then more recently, beautiful crystallographic work where they actually showed this in the crystallography, and they had DQ peptide T-cell alloreactive panels to actually show the recognition that there are two sorts of DQ molecules. DQ group 1 or G1 is any DQ alpha 2, 3, 4, 5, 6 can form a heterodimer with any DQ beta 2, 3, 4. They called group 2 or G2 molecules, any DQ alpha O1 chain could form a stable heterodimer with DQ beta O5 or 6. But you cannot form a stable heterodimer between a DQ alpha here with a DQ beta in group 1, and you cannot form a stable heterodimer between a DQ alpha group 1 with a DQ beta of group 2. So now, some of us have one molecule, some of us have two molecules, and some of us have four molecules, and it's entirely prescribed by the paternal and maternal haplotypes we have inherited and how much homozygosity or heterozygosity we have. And you can just follow this diagram to show how many molecules you might have. Consider now if you are dealing in a transplant, you're introducing a DQB1 allele mismatch into the transplant. There are some transplants that have a one molecule mismatch, some have two, and some are mismatched for 3 molecules, particularly if you introduce a DQ alpha mismatch. So we tested this hypothesis in unrelated calcineurin treated patients. G2 molecules increase the risk of relapse. We were able to show that when a G2 positive patient whose germline increases risk, if that G2 molecule is the mismatch from the donor, then they have the highest risk of all. This is the green group. So presence of the G2 increases relapse, and donor mismatching against that G2 elevates that risk. And here are the hazards of over 3-fold higher risk of relapse if you have a donor that creates a mismatch against a patient's G2. We were able to validate these findings in the haplo. So here are the data for haplo PTSI, G1, G2 patients transplanted from G1, G2, very high risk post-transplant. So now we've got a simple kind of a dimorphic definition for DQ, G1, G2, and the last group of slides is to tackle the accessory molecules. As many of you know, DM and DO play a really fundamental role in facilitating peptide exchange, and in the literature, DO is sometimes referred to as an inhibitor because it may modulate those effects. So they're all polymorphic. For the sake of time, I am not going to go over the negative data for DO, but the DM is very interesting. There are multiple residues that clearly are functional, DM alpha as well as DM beta. And when you divide up the DM into 2 groups of molecules, either a 101 DNA with a 101 DMB, calling that a DM11 molecule, they are functionally very different from DM molecules that are not a 101 or 101. Now you have a beautiful model where you can string DR beta 86 val gly, DQ alpha beta G1, G2, DM alpha beta 11, non-11, and now you can finally ask the all-important question: does the presence of certain extended class 2 haplotypes influence transplant outcome? And the answer is probably yes. And these haplotypes themselves confer very individual risks to the transplant recipient. So we've learned a lot through the years, And it's not all about simple donor match or mismatch. We are needing to think really well beyond that. Individual germline variation. What is the patient bringing to transplant? How do we use that to benefit the patient in selecting a donor that will lower transplant risk? So I hope I've shared some new information for you that we can all think to add to our armamentarium and tool chest for transplantation. Again, would like to thank all the members of the IHWG. And of course, thank you so much, Dr. Reed and the UCLA CTSI series. It's been such a pleasure to be here today to visit all of you. Thank you.
On. Yes, thank you, Effie. What a beautiful talk. I just enjoyed it. I always learn so much when I hear you deliver lectures, so I've learned so much. Thank you. Well, I'd like to take some questions from the audience. Please, Jennifer. Can you use the microphone? Hold on one second. It's coming to you.
I'm always fascinated by your research. I have a lot of questions, but I only ask one here. So for the B leader mismatch match and also the MICA, I think it's all relevant to NK cells. It seems like as a regulate NK cell activity. So I wonder if also you also look at the KIR mismatches, the activating KIR inhibitory, because NK, oftentimes we look on the one aspect of NK cell, but not the whole picture. So there's so many factors involved in NK activity. So particularly for cytophosphamide protocol, it seems like it's a T cell replete protocol, which T cell play a very important role. So I just wonder how important NK cells are in the haploidentical transplantation.
Really, thank you for that question, Jennifer. That's been a real high priority question for many, many laboratories. I would like to just give a nod to Dr. Catherine Hsu, who's collaborated with our lab for the IHWG on NK and KIR. So I'm sure you are familiar with a publication several years ago now in Blood from the Brown et al., where they looked retrospectively at haplotransplants receiving PTSI, and they could no longer see either a benefit or a detriment based on donor KIR. And this really spurred a lot of community interest and say, why? What? They're saying, we've lost the KIR effect. What do we do with PTSI? Well, I think it's because, think about what we're doing when we're administering day 3 and day +4 PTSI. So here you just infuse this huge volume of alloreactive donor T cells and NK cells that came in with the graft. Now you're then giving the patient PTSI, you're getting a total abrogation of all those alloreactive T cells and NK cells. And then within 3-4 days, you will begin to repopulate NK first before T, but those NK cells are actually not from the infused graft, as you know, and those NK cells have very, very naive phenotype. So if you think about it, there may be a biological reason why we've lost the KIR effect with PTSI is that we're totally shifting the T cell NK repertoire, and we have to wait months before those NK cells mature and are having the phenotype to be able to do that. So we and others have not been able to see a really pronounced KIR effect with PTSI in the haplos. And I'm just gonna, assume that when people are starting to give PTSI and unrelated, it's going to be the same because the action of that drug will be very, very similar in the unrelated as in the haplos. So it doesn't mean that the KIR has no role. It means that we're totally shifting the dynamics of what's happening in vivo with that drug. And we should not be surprised that certain correlations that were really strong before with cyclosporin or TAC are no longer seen because those cells really aren't there and aren't expressing that. Yes. That is a puzzle. So, naive, maybe you get more NKG2C/E earlier on. And then, but it's also, temporal in a way. So I think we still have a lot to know about the sequence of receptor expression as those NK cells start to mature. Yeah.
I have a question that I'm just fascinated by the promiscuity of DQ.
Oh,
Oh
really?
my goodness. So the question actually is going to go back to solid organ transplant.
Yes.
DQ is our biggest problem in transplant, causing chronic rejection. Almost all humoral immunity is against DQ. No one has really figured it out except to talk about polymorphisms. But I think your data may explain some of the findings because certain certain recombinations you might be seeing because of new alpha beta pairings.
Yeah.
I'm just wondering if you've given any thought to this potential problem.
I think Dr. Reed should do the study, don't you think? I think what you need,
it's a really great study to do.
I think what you should do is
Yeah.
you get your kidney donor,
Mhm.
DQ alpha beta
Sure.
characterized. You put together the alpha beta heterodimers, and we just had a paper accepted. It should be online, at least the online version. It may not be the pretty version, you know, but we have a tool. You plug in the DQA1, you plug in DQB1, and it gives you the alpha beta crisscross cis and trans. And so I think the hypothesis would be for graft rejection in solid organ is
Mhm.
figure out your patient alpha beta, figure out the donor alpha beta, and
Right.
how much is truly different between the donor and the recipient going host versus graft rejection vector. And tell me what you find, because the hypothesis would be that the graft rejection rate will be much higher and more brisk amongst those with 2 or 3 molecule mismatches.
And the humoral immunity, I think we're
And the
looking at
humoral-
alloreactivity,
absolutely.
right?
Because
Exactly.
your antibody, your anti-DQ
Yeah.
antibodies may also track.
Exactly. I think
with
they're
the
a good
right?
biomarker for alloimmunity.
Oh,
for that
yeah.
study. It's
Yeah.
really good. There is one question from the online audience from Suzanne Welsh. She's asking, from a healthcare equity perspective, the higher match probability of white Europeans is not surprising. But is there any explanation for the highest match probability for Native Americans, which has an underrepresented class of individuals, might seem surprising to me?
Yeah, I thank you for that question. That's the first question I actually asked Lauren Greger. It goes, how do you explain the Native Americans? And the only thing that we can come up with is they have really highly conserved haplotypes. So when you can match, you can match. And it's just like if you had A1B8DR3, you know you're going to get 10,000 donors like that, right? And then you can go to all your other secondary characteristics. And it just could be that in that particular cohort of patients who self-describe as Native American, that they had haplotypes, strong LD, and you know, it's all about LD in a way for HLA, right?
Agreed. I was
Mm.
thinking it was just.
Yeah.
You have a question, please?
Yeah, great talk. I was interested in terms of central tolerance and peripheral tolerance, how these mismatches can play a more favorable outcome. Do you think, and a lot of them had to do with like cytoplasmic domain or where the peptide binds. Is this something that you would describe as there's more wiggle room for tolerance to kind of not be so strict? And that's what allows better outcomes, or what? I just want to know your thoughts on that.
Yeah, it's really great. And this is where, you know, these association studies, they start us off in, you know, testing and putting on the table potential features that can give us clues into mechanisms. But now the big key is, how does this happen? How do you explain that a mismatch is favorable? I've asked this of Ephraim Fuchs many times. I said, so Ephraim, how did you explain the Hopkins data? Because you showed it first. They were the first group that showed that DR beta mismatching was actually protective. That was Hopkins data, and they had very robust numbers even then. And, you know, Ephraim is still working on that, as are we, because we've now been able to replicate that in multiple different cohorts. So we know it's real. It's not a fluke. It's not specific to just one center's data versus another. And I don't have an answer for you, but it does. I mean, we're at so much at the early stage now. And I think what we've learned is that it may not be as simple as maybe we thought we were dealing with, is because now you have so many things to consider in the allo response and transplantation. So it's not just the donor, it's also the patient. I didn't even cover the wealth of literature now in terms of what CMV does to these receptors. Okay, so you will get a profuse upregulation of expression with CMV. And so patients who are CMV seropositive who reactivate are maybe a very interesting group to look at. And we don't know because CMV just sends out all these decoys, right? And the HLA glands onto them. And that really may impact how these NK receptors are actually surveying and being able to either mount an activating or an inhibitory effect. So I wish I had the answers, but I think it's going to take all of us, you know, with very different approaches and different mindsets to really chisel away at it.
Can you hear me? Okay. So thank you. Very nice talk, and we're learn a lot from publications and even today the lecture today. So my question is, since you know the third generation sequencing will be almost ready for the HRE sequencing. So the length of this such as nanopore sequencing will be about tens of kb, and my thinking that can we just moving on the haplotype sequencing for the whole HR gene, whole HR area or region, I mean. So do you think how far we from there? Because if because if we got haplotype for class 1 and class 2 as well, probably have more big picture for
Yeah.
mimic this model or outcomes, and not only the class 2 or something, the haplotype, right?
I think we're inching our way there. Still the next gen platforms that are out there commercially, they do finally allow us to string exons, introns, and possibly regulatory regions, 5' promoter, 3' UTR together. Hopping from gene to gene, I think, is going to still be a huge problem for us. And you probably know, class 3 and class 2, you've got all these hotspots of recombination. And what that does is it just disrupts it. And we still don't have a good idea in terms of at a haplotype level, how those recombinatorial hotspots are then breaking up this. And it could be very different by haplotype. So, yeah.
We're going to take the last question here.
Thank you for a wonderful talk. This question is following to Dr. Zhang's question earlier. So we know that the B locus is highly variable, but we see consistently from your data that methionine and threonine are protected. So do you have any insights why those genotypes are so protected, molecularly speaking?
You mean the leader peptide?
Yes.
Yeah, it may have just the fact that in my mind, there are probably two ways to think about this. Since you have such tight conservation of methionine or threonine to the actual B leader antigen domain, it could be that the leader may simply be a proxy for the peptide binding domain. So we are pursuing the peptide binding domain and the repertoire with machine learning and things like that. An alternative mechanism is that in addition to its peptide presenting function, HLA-B may be influencing HLA-E, and many labs have shown higher or lower ability to get HLA-E stably expressed at the cell surface, which then HLA-E then has its dual function, right? It can easily present peptides to T cells, and then it can also serve as a ligand for NKG2. So there are probably a minimum of 3 different pathways that can be used to explain this differential effect of that leader dimorphism. But great question. Thank you.
Okay, well, we'll close this session. And again, thank you so much for the wonderful presentation.